The sound of Mozart, the sight of a beautiful painting, the touch of a mother's hand, the scent of a beautiful woman, and of course, the taste of foie gras. These are our senses, and their role is to create an internal representation, a map, if you will, of the physical and chemical features of the outside world. Now, what is not generally appreciated is that our view of the world, what we capture with our senses, is only in our minds. Can you hear me all right? For example, we see in color because we have photoreceptor neurons in our retina which are tuned to red, blue, and green, which together cover the full visible spectrum. In contrast, as it turns out, dogs don't have red pigments. So the view of the world that dogs capture is completely different from ours. Here you see 3 cute little puppies, the way we see them, and here are the same 3 puppies, the way they see themselves. So you could see that in essence we create our own selective image based on our own individual sensory inputs. So in essence then, the brain functions not by creating or recording an exact picture, but one that we individually construct. In fact, every species lives in its own sensory world, of which others might be partially, or in some cases, completely unaware of. Now, what I'll do now is tell you about our own work looking at innate behaviors as a strategy to decode the magic of the brain. Now, innate behaviors are behaviors that trigger predetermined actions. And this could be in response to an external sensory cue or in response to our internal state. An example of innate behaviors are our attraction to sugar. We have no choice but to love sugar. And this is true in every animal species. You might learn not to like sugar, but we're all born with a hardwired attraction for sugar, and conversely, with a hardwired aversion to bitters. These are examples of innate behaviors. I have a horrible echo. Is the volume way too high? It is. Can we lower the volume, please? Hello, can you hear me? Can you hear me now? Can you hear me now? Good, we'll keep it there. So, the example of sweet and bitter are instances of innate behaviors triggered by external sensory cues. But we also have innate behaviors which are activated by our internal state. And one of the best examples is our sense of thirst. This is the motivation to seek water and drink when we're thirsty. Now, we reason that, in fact, then there should be an area in your brain where you represent the sense of thirst. And this might be your brain circuits that control our innate drive to drink when thirsty. So we set out to look for brain areas which are activated when the animal is thirsty. And much to our delight, we identify a small nuclei at the center of the brain with only a couple of hundred neurons, which are robustly activated when the animals are very, very thirsty. And you could see the neurons in green And what you're seeing in red in here is the activity of the neurons. It's a marker for neuronal activity. So we make the mouse very, very thirsty. These neurons are screaming. There are 100 million neurons in a mouse brain, and around a couple of hundred of them are robustly signaling. Now, if these neurons represent the sense of thirst, there is one prediction, a thought experiment. We should be able to take a mouse, magically go into their brain, activate these neurons, and this mouse will think it is thirsty, irrespective of its internal state. Agree? It turns out that there is technology to do just that. And this was developed in part by the third speaker today. And this is a family of proteins, ion channels, that we can target to any neuron we want. And using light, we can now turn these molecular tools on. So we can take a mouse, introduce these light-activated ion channels, that's what they are, put a fiber optic in the brain of the mouse, right above where these neurons are. Now turn the light on, and now what we'll do is activate only this group of neurons from the 100 million neurons in the mouse brain. Now what you see here, you're going to see a mouse that's been completely water-sated. This animal has been giving water for the last week. He has zero interest in drinking water. It's in a cage. Here's a water spout. And you're going to see a fiber optic cable going into the brain of this mouse. And this mouse spotting around, goes by the water spout, no interest in drinking. It goes off to the side where we have some toys. Now we turn the light on, and the animal zooms straight into the water spout. And the animal drinks and drinks and drinks and drinks until we turn the laser off. It ceases all behavior, and it goes back to play. You can now turn the light back on, and the animal goes back to the water spout and drinks and drinks and drinks and drinks. This animal will drink 15% of its body weight in water in around 15 minutes. This is what controlling innate hardwired behavior is all about. Now, most of our work, it turns out, is not on the sense of thirst, but on the sense of taste. And we use the biology of taste to get into the window of how you transform detection into perception by our brains. Now, over the past few years, we have identified the cells and the receptors mediating all 5 basic taste qualities. We have only 5 basic taste qualities: sweet, sour, bitter, salty, and umami. Umami, well known in France, is a Japanese word that means yummy, delicious, and it really represents savory taste. It's actually the taste of proteins and best associated with mushrooms, cheese, seaweed, and so forth. So that is our taste palette. Now, over the last couple of years, we've also identified receptors for carbonation. And this explains our capacity to both enjoy and seek the bubbly in champagne and carbonated drinks. Now, our work has shown that each of these receptors, mediating the 5 basic taste qualities, is actually expressed in unique classes of cells in your tongues. These are your taste sensing cells in the tongue. And the unique aspect of the organization of the taste system at the periphery is that each taste quality is expressed in its own cell type. So what you have in your tongue are cells that respond to sweet, cells that respond to sour, cells that respond to bitter, salty, and umami. And most critically, each of those function as a label line all the way from your tongue to your brain, just like the keys of a piano. Now, to illustrate how you can manipulate these label lines in your tongue to dramatically alter taste preference and perception, I'm going to show you one experiment. It turns out that mice, like people, enjoy the good life. So if you take a mouse and you give a mouse a choice to drink from a bottle containing water or a bottle containing water plus sugar, this mouse will prefer 50-1 the sugar versus the water. Well, we developed the means where we can actually measure this scientifically. So we train mice to go into a little testing chamber. One of 8 to 16 different bottles goes into place. And now the mouse is being trained to lick from a little spout. And we have a little laser beam that counts the number of licks. And it turns out that the number of licks the mouse makes in a short window of time is a perfect indicator of its preference. So they lick for 5 seconds, one solution, random, then a new one moves into place, and then the animals lick. And depending on how much they lick of each of these random solutions, we can now deduce what its preferences are. And for example, if we test the mouse by giving him different concentrations of an artificial sweetener, the more you give of saccharin, the more they lick. I think I have a little video that shows the behavior. There is the mouse. There the little bottles move into place. There is the little window that opens. There the mouse licks. Little laser counts the number of licks. And now we know what the mouse likes. And when you do that with a wide range of sweet-tasting chemicals, you find an unexpected surprise. And that's the fact that even though they enjoy many of the sweets that we do, looking at the left side of this panel, like sucrose and saccharin, it turns out that mice are completely insensitive to the most prominent of all known artificial sweeteners in the Western world, at least, which is in the example here, aspartame. So we reason that perhaps the reason that mice cannot sense aspartame like we do is perhaps due to underlying differences in the corresponding taste receptors, differences between mice and humans. And so we did the obvious experiment. We went into the human genome, identified the human taste receptors, took the human sweet receptor, and now genetically engineer a mouse that expresses the human receptor. And lo and behold, we completely humanize its taste responses. So we have now a range of mice that in essence innately like the same sweets, similar bitters, umami, and so forth as humans do. I think the purpose of these experiments is not to play with a mouse brain, but to uncover the logic by which taste is encoded and decoded at the periphery. So then how does the tongue know what it's tasting? It's very simple. All it needs to know is which cell is being activated. Now, what about the brain? How does the brain know what the tongue knows? Well, it's been known for a long time that each of our senses is represented in a unique area of our cortex. And so we have an area dedicated to sight, an area dedicated to sound, an area dedicated for touch, and we also have an area dedicated to taste. And this is known as the insular cortex, which I think is highlighted there with a little circle. So we developed a handful of years ago a strategy to actually image the function of a mouse brain in vivo, alive, while the animal is performing a task. And in essence, what we do is we create a little window into the animal brain. So we make a small craniotomy around 1 millimeter. We know where we are in the brain because of the vasculature. This is not like a fingerprint, but it's good enough to know where you are. We introduce into those neurons in that area of the brain unique sensors of cell activity. These are molecular markers that change their properties when the neurons fire. And then we put this live mouse under a unique imaging microscope that allows us to see what the brain is thinking when the mouse is tasting different chemicals. And when we did that by looking at bitter and sweet and umami and so forth, we came up with a rather unexpected discovery. And that's the fact that each taste is organized in your brain as a unique, segregated cortical field with, in essence, each different taste creating a map of taste qualities. So you have an area of your brain that is dedicated only to sweet, and a different area in your brain that's dedicated only to bitter, and a different area only to umami, and so forth. So then, how does the brain know what it just tasted? Well, all it needs to know is which area has been activated. Now, if this is true, and these cortical fields in your cortex represent taste, there are two predictions. And because of time, I'm only going to demonstrate one of them. The first prediction is that if we could somehow go into your brain and silence, inactivate one of those cortical fields, let's say sweet, I should completely abolish your ability to perceive sweet, even though I'm giving you sweet in the tongue. Agree? And that's exactly what we've done. The second prediction, if these areas really represent taste, is that we should be able to go into your brain and activate one of those cortical fields. And you will think you are tasting that taste, even though I'm giving you nothing. And let me show you now what bitter tastes like to a mouse. All right? So here we use the same tool, optogenetics, where we introduce one of these light-activated ion channels, but now we activate it only in the cells that represent bitter. Then we put a fiber optic into the brain of the animal. Now we give the animal only water to drink, and in some of the trials, we activate the bitter cortex. And let's see what the animal now perceives. So you're going to see here a mouse that's been trained to lick from that water spout when a little yellow cue comes on the top. This is a very thirsty mouse, and so it has two cannulas in the brain, one going to the bitter center, and you could see that the mouse, every time the cue comes, it licks the water. These were 3 controlled trials only with water. This is now the 3rd trial. And this is what a thirsty mouse would do. Now we're going to activate the bitter cortex when the animal is drinking water. First trial. Second trial. Third trial, a little bit more light. And the last one is a lot of light. This is an extraordinary finding. This mouse is cleaning its mouth of a fictive taste. This mouse is drinking nothing but water. But in essence, what we have done is demonstrate the ability of our brains to directly recapitulate sensory perceptions and behaviors which are naturally indicative of orally provided tastings, but in this case, in the total absence of any sensory input. So taste, in essence, is just in your brains. Now, let me finish my talk with the Mary Poppins experiment. If you remember, Mary Poppins said that the spoonful of sugar helped the medicine go down. So we reason it should be possible then to design an experiment where we give the mice in the tongue something completely unpalatable. Let's say a bitter taste, as shown in the diagram here on the right side. But in some of the trials, what we're going to do is when the mouse is drinking that bitter, we're going to directly activate the sweet cortical field. And if we are correct, we should be able to transform an unpalatable taste into a palatable one. So this, in essence, is the experiment. And we rig the system so the animal's licking automatically activates the laser, so that the animal itself is driving its own behavior. And so here's an example in which we have 15 trials. And 11 of the 15 trials, the animal is only exposed to the bitter chemical. And you could see the animal is barely licking each of those bars. And in 4 of the trials, we put the laser under the own animal's lick control, where you now activate sweet cortex. And much to our delight and surprise, we have transformed something horribly bitter into, in essence, honey and ambrosia. Okay? So I think these kinds of experiments beautifully illustrate both the power of innate behaviors in driving these circuits, but I think more important, it also shows how by directly accessing our brains, we can completely change our internal representations. Now, I have 30 seconds left. So let me conclude by saying that understanding the human brain is the greatest scientific challenge facing our generation. Now, we are at the beginning of an extraordinary journey into the center of our thoughts, memories, and emotions. Now, our brain, the human brain, has 85 billion neurons. And together they make over 1 trillion connections. This level of complexity is nearly incomprehensible. But I believe, thanks to great advances in biology and technology, we're getting a window into the inner workings and the magic of the brain. I hope in this short 19-minute presentation, you've had a flavor and a feeling for the magic of the brain. Thank you.